Sì, tudo bem, sim. Okay, good afternoon, everyone. Thank you for being here. My name is Juan Pablo. I'm here in behalf of Brain Support Corporation. I'm going to make the presentation, an introduction for the NIRS acquisition system. And in this presentation, we are going to talk about an introduction to functional near infrared spectroscopy. That's what we are going to do in this part. After that, we are going to do the live demonstration, a hands-on that you can watch on the online, and then we can have an open discussion and closing remarks. Uh, just 1 second, please. You can share this full screen. I think
Gabriel pergunta. Aparece aí no caminho. Esse é o outro pergunta.
Mas é a tela que está.
Não está conseguindo compartilhar, não?
Compartilhar em tela cheia. Se compartilha tudo
se for apresentar e apertar.
Tela cheia, antes de apresentar assim.
Ctrl Tab, Gabriel.
Como?
Ctrl Tab, tu consegue voltar para a tela do tela e aí compartilhar, assim, já está na apresentação. Ctrl Tab.
Assim. Não, deixa eu botar a tela cheia. E se compartilha a tela toda?
Está aparecendo
Tem. Será certo?
Coloca na apresentação. Coloca na apresentação. Ativado.
Quando vai para lá e sai da tela.
Aí está compartilhando.
Não, aparece. Isso.
Isso.
Agora vai.
See?
Sí.
Thank you. Thank you. I'm sorry for that. We had some technical issues, but we are back on track. Okay, so this presentation we're going to do a review on the basis of the near-infrared spectroscopy. Um, as we already saw some really good presentations on what we can do with the NIRS, with what are some of the research, what are some of the topics that you can research with NIRS. In this case, I think it would be good if we go into the physiological aspect of the near infrared spectroscopy so we can understand what we are measuring, and based on that, how can we create the designs of our experiments. So, what is NIRS? NIRS is a cost-effective technique, a neuroimaging technique that is robust. Why is robust? Because it's based on physiological understood aspect, that is the ball response. We are going to see that later on. Is flexible. It's a flexible technique because you can answer many questions about with NIRS. You can answer educational questions. You can answer cognitive questions. You can answer even in sports. People use it to do research with the NIRS. So, what is the physiological basis of the NIRS? The NIRS is going to record the neurovascular coupling activity in the brain. So, as we can see here in the image, we have two moments. We have a no stimulation baseline activity in the brain. So, we have a normal current blood flow. We have the capillaries and the arteries, and the red dots are the hemoglobin with oxygen, and the blue dots are the hemoglobin without the oxygen. This is the blood flow normal in a resting state. But when we have neuronal activation, when we have the presentation of an stimulus, the neurons are going to need more oxygen. So to do that, what happens is that the current blood flow rises, the capillary is going to be bigger so that the hemoglobin can transport more hemoglobin with oxygen to these areas. But how does it do it? It is important here that you understand that the hemoglobin with oxygen is going to be higher than what the neuron actually needs. Why is that? Because the brain is not going to send just the precise amount of oxygen that the neuron is going to need. So as you are going to have more hemoglobin with oxygen in this part, then you are going to have less hemoglobin without oxygen. In this case, we are going to talk about the oxyhemoglobin as the hemoglobin without oxygen, and oxyhemoglobin as the hemoglobin with oxygen. Just so you can, so that we can keep on track on that. So oxyhemoglobin, after you present a stimulus, is going to rise because of the current blood flow. It's going to be bigger. The neurons are going to use more oxygen, but the brain is going to have more blood. So the oxyhemoglobin is going to be less than what you have in no stimulation baseline. So what we can see, how does that look basically in real life? The oxy and the oxy have a color property. When you have hemoglobin without oxygen, it has a bluish color, as we can see in the baby in the left picture. And the oxyhemoglobin with the oxygen is going to have a red color. That's why we have the cheeks red in the baby. How then this color is going to affect what we are going to use in the NIRS system. That's the absorption of oxy and deoxyhemoglobin of light. So as we can see here in the figure, we have an absorption factor of light in the y-axis, and we have a wavelength in the x-axis. The property, when you send light to tissue, it can be absorbed, it can be scattered. We are going to see a figure on that later. But when you have hemoglobin with oxy and deoxy, this absorption factor is going to be different. You also have absorption factors for water, but in this case, we are interested in the part in the optical window that is around 700 and 900 nanometers. As we can see here, the 700, then just a minute. To discriminate between oxy and deoxy, the NIRS system uses multiple wavelengths in measurements. In this case, and in general for the NIRS system, you are going to use a wavelength of 760 nanometers and a wavelength of 850 nanometers. Why? Because as you can see in the figure again, in the 760 nanometers, we have a peak for the deoxyhemoglobin absorption factor. And for the oxyhemoglobin, we have a peak in the 850 nanometers for the absorption factor of the oxyhemoglobin. So usually the systems are going to use these two wavelengths to discriminate between deoxy and oxy. So when you have a recording from NIRS, you are going to have a wavelength that is going to be absorbed by the deoxy, and the other one that is going to be absorbed by the oxyhemoglobin. Now, why does this work? So if I send a near-infrared light, the near-infrared light is going to be around this window of wavelength, is going to be based on the Beer-Lambert law. What does the Beer-Lambert law say? As you can see here, the absorption is going to be proportional to the concentration of the sample. So if I have more oxyhemoglobin in one area, the absorption of the light is going to be bigger as if I didn't have that much amount. It's going to be the same for the oxy and the deoxy. So basing on what the picture that we had before, this one, when you have no stimulation baseline, you are going to have less absorption of the light light in this situation or in this context. But when you have neuronal activation, you are going to have more oxy, less deoxy, and in that case, you are going to have more absorption for the oxyhemoglobin. Now, what does the light usually do? The light doesn't only get absorbed. The light can have different processes when it's going through a tissue. It can have transmission. That is, if I send the light in a certain straight line, for example, it can appear in another part of the tissue. That is what we call transmission. We can have reflection. Reflection is basically you send the light through the tissue, but it's not going to pass. It's going to be reflected. We have a scatter. Scatter is when the light and the photons of the light are going to be spread around the tissue, and we have absorption. All these aspects are going to be important for the NIRS system because these are some of the aspects that we use to transform light intensity to concentration of hemoglobin and deoxyhemoglobin, oxy and deoxy. Now, how does that look in a NIRS system? In a NIRS system, we have an emitter and we have a detector. The emitter is the one that is going to be sending the light through the skull and through the brain, and we have a detector that is going to detect the light that is going to be sent by the emitter. In this case, the sensitivity profile light conforms to an elliptical banana shape as it travels from source to the detector. This is known as a photon banana, as we saw before in a presentation. This is going to be different for every distance. So if you have a distance of of 3 centimeters from emitter and detector, you are going to have a you are going to be able to record data from the brain. If you have less, then you are going to be recording more skull activities. So, and if you have a bigger distance, then you are going to record more brain activity, but the light is going to pass through other processes. It's going to be scattered more, it's going to be transmitted more. So 3 centimeters is what is the standard right now. If you have any theoretical assumption on why you would want to have a different distance, you can do it, but the standard in this case is 3 centimeters because it's the appropriate distance that it allows you to record neuronal activity, right, in the cortex region. That is one of the differences that with the fMRI technique. The fMRI technique, it's a technique that is going to give you more information, more local information, and it's going to be in more deeper areas of the brain. In the NIRS, this is one important part for your research, is going to give you information on the cortical surface of the brain. So usually if you have a question in your research that is going to refer to a more deeper region of the brain, it won't be useful in your, the NIRS is not going to be that useful for you in this case. So what is the response that you are going to see based on this hemodynamic or neurovascular coupling that we already saw? This is important, as I already told you. The NIRS is going to be, as it is based in the neurovascular coupling, the timeline or the time response, the activity that you are going to see is going to be longer than, for example, a person that works with EEG is going to be it's going to be longer than EEG activity and more. It's going to be more alike with the fMRI activity. So when you have the stimuli, let's say here in the timeline in this graph, you have 0 as the presentation stimuli, then you are going to expect that the peak of the activity for the hemoglobin response or the hemodynamic response is going to be around 6 seconds. You are going to have and then it's going to go down at around 8 to 10 seconds, and then it will go back. It will go back to baseline at around 60 seconds. This is a subject that is usually really discussed in the articles, but this is the hemodynamic response standard, and it is usually based on the fMRI studies. As we can see, the oxyhemoglobin, as I already told you, based on the first figure that we saw, is going to, the neurons are going, the activity or the hemoglobin is going to have more oxygen, and that is why you have a rising in the oxy in this graph, but it's going to have less deoxy. As you can see here, in the window of 4 to 8 seconds, you are going to have less deoxy. This is weird because as you have more oxygen, more hemoglobin with oxygen, it will be intuitive to think that you will also have more deoxy because the neurons are going to be using that oxygen. And that is why I told you that the oxy is always going to be bigger than the amount of oxygen that the neurons are going to use. That is why you have this behavior in the activity of the oxy and the oxy. So after you have the response, then the deoxy is going to be back to baseline concentration. This is important not only because this is important mainly for your stimulus presentation periods. You can have, for example, if you have, for example, a study with EEG, the stimulation period can be around 1 second. if you had a presentation of 1 second for your stimuli, the hemodynamic response is not going to be at its peak in that moment. So when you have studies with NIRS, you are going to need presentation stimulus of at least 8 seconds, 6 seconds, or you are going to need a block design where you have many stimuli of the same characteristics and analyze the activity on that block design. I don't know if there is a question here. I know this is kind of difficult because it's the dynamic response. No, there is no question. No. Okay. So what is once we understand the hemodynamic, the basis of the function, the physiological basis of the NIRS and what is detecting, we have to understand what is the topography of the NIRS. The near infrared light detected through a medium from a single light source. So you are going to have detectors and you are going to have sources. The source is the one that is going to be sending the light, and the detectors are the ones that are going to detect the light. And you can have more than one source sending light through to many detectors. So you can have one source, as you can see here in the figure, and you can have two detectors detecting this light. The source to the detector pairing distance affects the depth of the photon banana penetration. As I already told you, 3 centimeters is the standard. If you have less, for example, in the systems with short distance channels, you have a detector that is going to be 1 centimeter. The distance from the source to the short distance channel is going to be 1 centimeter. So in that case, you are going to detect the difference that is going to be in the skull. So in that case, you can use that information to eliminate it from the signal that you need, because you only have the noise in that short distance channel. So if you use that to eliminate it with the signal that you have from your source and your detector, you can have only the neuronal activity. That's why they have this short distance channel NIRS. One data channel is produced per wavelength used per source detector pairing. What does this mean? As in EEG, for example, one channel is one electrode. In fMRI, you can talk about voxels. Here, the channel is the pair of the detector and the source. So here we have two channels, and we have two wavelengths sent for those two channels. So in this case, we will have four channels. So you have source, the source and the first detector with two wavelengths, and we have the other detector with the source, that's one channel with two other wavelengths, and that will mean that we have here 4 channels. Why is that important? Or why is it like that? Because you have the oxy for one pair and the other pair, and that is going to be analyzed differently for each wavelength. As the nearest neighbor measurements are done with a spacing of 2-3 centimeters between sources and detectors. Here you have a source, you have a detector, and you have a data channel. Excuse me, I think we have again a technical issue.
Yeah, just a sec. Outside. Yeah, go there. It's okay. Just something.
Yes.
Sorry.
Thank you. Is it okay? No?
Not yet. Okay.
Okay. So in this case, as you can see, you can have as many channels as you want. Well, that will be based on your NIRS system. But for example, in this case, we will have the source that is in the middle that is going to be sending light to other 4 detectors, and that will be 8 channels because you have 2 wavelengths for 4 detectors, and that will be 16 channels in this case. So that will be one of the main ways of placing the sources and the detectors in one single area or region. What are some of the examples of how NIRS is used today? We already had some thoughts about it. You can use hyper scanning, that will be multi-subject measurements. So you can have the NIRS, for example, the NIRS port, that is the one that we are going to use today for the hands-on here in the university, is portable. The portability of that NIRS system is going to allow for you to use it in more real life situations. In this case, you have a recording for the person that is playing the violin and the other person that is playing, I think, a piano. And you can have the recording of both of them at the same time and see how the activity of the brain for those two is going to be synchronized. That is what we call is paper scanning, and it's usually it's easiest to set up than what you will have to do with the EEG. We can have ecological environments. I think this one was also used in the previous presentation. Here they recorded the NIRS for a person that was riding a bicycle. They proved the viability of the portable solution of the NIRS. So although you have a movement noise, it can be filtered and it can be filtered and it can be solved, this type of noise. So you can have a good signal and you can have a good experiments with this type of design. You can also have the triggers. The triggers are usually one of the biggest issues in the laboratories when they are starting their experiments and with the NIRS. The NIRS systems have a portable manual trigger system that will be, for example, usually the triggers are going to be sent through a program. But if you have a real life experiment that the person is outside, they can send their own triggers to the software, and that is going to give you more information on what the person is doing, where it is seeing something that they like or interacting with another person. Those things can be triggered in this case with a manual trigger that the NIRS has. There is also the opportunity to make experiments with infants. It is child friendly. It can increase the increased interest of the researchers. Right now it's bigger because it's easy to place, and the surface of the skull for the baby is usually tighter. So that will allow you to record more neuronal activity for the babies. You can have a more complex stimulus, object processing, social communication, biological motion processing, face processing. So for the infants, it is usually a really nice technique to use. So how does the NIRS compare to more well-known functional brain imaging modalities? The comparison between the fMRI and the NIRS would be the fMRI, as we are going to see later, is going to analyze the relationship between the oxy and the oxy, while as the NIRS identifies the oxy as well as oxy. They identify it separately. Usually for the fMRI, you have the relation between them. The fMRI has a restricted environment and the NIRS has a relatively small form factor, even portable as the NIRS port. It is a noisy operation, the fMRI. The NIRS has quiet or silent system operation. The fMRI has excellent spatial resolution while also measuring the whole brain. as the NIRS has good spatial resolution, but only of the cortex, just below the scalp. That is why your questions, if you're using NIRS, should be based on activity in the more cortical region. The fMRI has relatively poor temporal resolution. The NIRS has relatively high temporal resolution, but it is 6 seconds. That's why when we talk about NIRS, we usually place it in the middle between fMRI and EEG. The fMRI is sensitive to motion artifacts, and the NIRS is robust to motion artifacts, and we already have some good algorithms to remove most of that noise. The fMRI, one of the things as we already saw, the NIRS and the fMRI are based on the same vascular coupling. So this study was validating the activity of the NIRS and the fMRI, how well related were those activity, and the study estimates activation structures, and as we can see here, they were recording the left central region of the brain. And in the graph, the zeta score activation changes. It's quite related between them. Yeah. So that is what that's telling us is that the NIRS is basically working, and it's basically recording the activity that we usually were recording for the fMRI. It is quite related. New insights into brain function by overcoming some of their intrinsic limitation. That's what we were talking about right now. The NIRS, although it is related to the activity for the fMRI, has another advantages that it's the more realistic experiments that you can do with it. Now, what is the relation between EEG and NIRS? The EEG identifies cortical activity via action potentials. The NIRS identifies it via hemodynamic response. The EEG has excellent temporal resolution. The sampling rate is usually higher than 2000 Hertz. And the NIRS has relatively lower temporal resolution. The EEG measures very quick patterns related to action potentials. That's what we were talking about sooner. The stimulus presentation for an EEG can be slower than 1 second. For the NIRS, you usually will need at least 3-4 seconds, as we already saw for the hemodynamic response. The EEG has relatively poor spatial resolution, several centimeters around the head, and the NIRS has greater spatial resolution than EEG. It's around 1 centimeter. And the EEG is highly sensitive to motion artifacts. The NIRS, as you can see, has better noise performance with movements and muscular use. The NIRS can also be used with other systems, for example, the transcranial magnetic stimulation. There is no interaction between the magnetic field of the TMS and optical signal of NIRS. There is no artifact, there is no interference between them. It has an in vivo assessment of cortical activation patterns during transcranial magnetic stimulation treatment sessions. And the optimal location of the probes can be identified by means of single pulse TMS. So it is quite flexible, as we were talking at the beginning, but it does have some limitations. The progresses of NIRS over the years are also attributed to the giant development of NIRS methodology and data analysis to deal with. It has some well-known limitations. It only measures only cortical activity, has low relative temporal resolution, and it has low relative spatial resolution to EEG and to fMRI. That's why we place it in the middle. So we can do some things to avoid problems with false positives. You can have careful experimental designs, avoiding tasks with a strong systemic activation, avoiding additional stress and discomfort for the subject. You can have extra short channels. That's to use to regress extracerebral layer. That's what we're talking about before. You can have appropriate statistical or signal processing methods, independent component analysis, principal component analysis, adaptive filtering analysis with general linear model. In this case, it's harder for the researchers that usually work with EEG, because these are some techniques that you can see more in the fMRI research. It has multimodal monitoring. You can have multimodal monitoring. That's measurements of signals from the systemic physiology, skin conductance, blood pressure, and you should be recording or assess the area that you are interested. Why do I say that? Because you have 8 sources, 8 detectors, for example, in this system, the NIRS port. So you can record the whole area, the whole brain. So you are going to have a strong hypothesis of what is the region that you are interested. So you can have a good a good study. So for example, if you study is with motor area, you are not with motor imagery, for example, you're not going to be recording the occipital or the frontal. You usually are going to be recording the motor region. So in summary, the NIRS technology offers compact and low-cost sensing hardware. It's not invasive and it's a continuous measure. It's highly configurable and scalable. It is highly configurable and it has scalable platforms. It records the ball light response without the magnet. It has higher temporal resolution compared to the fMRI, but it has higher source localization compared to the EEG. Has minimal sensitivity to movement artifacts, and it is multimodal possibilities. It has multimodal possibilities for your research. So I think that will be all. I don't know if you have any questions on what I told about the NIRS. What does it record? No? Okay, I think that will be all. Yeah.
I do have a question. You said that fNIRS has an increased temporal resolution with respect to fMRI.
To fMRI, yes.
I was just trying to understand why. Because the phenomenon is the same.
The phenomenon is the same, but the fMRI records the hemodynamic response based on the magnetic properties of the response. So I am not an expert in fMRI, but what I do understand is that the fMRI generates this magnetic field, and that magnetic field is going to alter the spin of the water, and that is going to change based on the of the oxy and the oxy. But that response is going to have lower temporal resolution than the NIRS, because the NIRS is sending the light faster because it has a higher sampling rate. So you have a more, the transmission of the signal is faster in this case. Yeah, for the NIRS than for the fMRI. That's what I understand. Of course, I am not an expert on fMRI, that I won't be able to answer you with a more precise answer than that, but I believe it's because of that. I don't know, Daniel, you also had a question? No?
I'm not, I'm not interested when you said
Before? This one? Yeah. No? Yeah.
When you say the temporal resolution of fNIRS, and could you say how it would compare to the one on EEG?
How does it compare with what?
Because in EEG you have a number there, right?
Okay. That's the sampling rate. The sampling rate for the NIRS is 7.45 hertz. Sampling rate for the EEG is more than 2000 hertz. I didn't put a number in the NIRS because you can also change the sampling rate in the system, but the standard sampling rate for every source and detector is 7.45. We can see that later on in the hands-on.
You can change the sampling rate.
Yeah, but that will affect the speed of how does the light is going to be sent, and that also will affect the response that you are going to have in your recording.
Okay, click on the hands-on, we can see
Yeah, that is
what are
specific.
the options for the sampling, right?
Yeah.
And this is good for experiment with time reaction.
fNIRS is good for what, sorry?
Time reaction experiment is good to
Time reaction.
The
yeah.
reaction time.
Go test.
Yeah. So in that case, for example, because for example, a go-no-go task, an oddball task, you usually are going to present the stimuli faster. Yeah, the oddball tasks are usually used in the EEG technique, but you have a stimuli that is going to be presented 1 second, for example, and a cross fixation of 500 milliseconds, for example. But so in that case, the time of the stimuli is going to be shorter than the response of the NIRS, as I showed you in the graph. So what you can do, if for example you are going to make an oddball design, is to have blocks. So for example, if you have an EEG and NIRS study, what you usually want is to have a stimuli that is going to be presented for shorter times, 1 second, 2 seconds, but the stimuli has to be um, comparable between them in one block. So then, for example, you can have a stimuli negative, positive, and neutral, and you're going to present 30 stimulus with negative images, positive images, and neutral images. So what you're going to do there is analyze with EEG the stimuli, the single stimuli, for example, every image that is going to be presented. But for the fNIRS analysis, you are going to analyze the whole block activity. In that case, you are going to analyze P300 for the oddball and analyze the activity of the NIRS for the positive, negative, and neutral uh, stimuli that you are going to present in that block. Yeah, that is why it is so hard, because the devices exist, to have them, but the important part in this case is have a good design for your experiment. That's the biggest issue and the reason why you can be published or not. Your a really good design that you can analyze and have a really good argument of why that works with fMRI, NIRS, and EEG or TMS or whatever you're using. Yep. Any other question? Mm, okay. Thank you again.
Great. We just had the talk of Juan. Now we are going to have the talk of Professor Dráulio. He's a professor at the Brain Institute. He is going to talk about the fMRI and the bold signal. Dráulio, please. Dráulio, you can come here.
A apresentação é muito fácil.
Oh.
A número de foto. If you want, you can conectar ele no Wi-Fi e entrar no streaming. Mas é Keynote. É Keynote. Não é focal. Vai ter vídeo também.
We're just preparing. the presentation. Just a few minutes.
So, good afternoon, everyone. First, I'd like to thank Brain Products for the partnership with the Brain Institute, understanding that this will be a very interesting site for human research, as in effect that we have been doing fMRI, EEG, and now the possibility of doing fNIRS. And you know, I'm happy to see that this will also increase the collaboration of the Institute with many other departments in the university. So thank you, Brain Products, for the workshop. Thank you, Brain Products, for the partnership. So the idea here that Daniel asked me to do was to present the basis of fMRI and maybe trying to understand what are the links between fMRI and fNIRS. I believe that the main link, and that's where I'm going to be concentrated at, is that fMRI is, like I was asking Juan right now, fMRI uses the same kind of phenomena that fNIRS uses. So part of my talk today would be to describe the details of that phenomena that we call blood oxygenation level dependent. I am not sure how deep did you guys go into this phenomena today. I'm not sure if you had a previous presentation on BOLD and fMRI, but anyways, let's move with the plan. So the idea of fMRI comes from what we call in general functional neuroimaging. When we started doing imaging, the advances of doing human imaging was definitely a great milestone in general, because you were able to understand or to observe different organs without the need of opening the body. So the first techniques, the first imaging techniques, including MRI, they had the objective of observing the differences in anatomy. So differences in anatomy of different organs, and then trying to understand if that organ was normal, if that organ had some kind of strange object or any change in the anatomy. What happened with these other techniques is that if you want to do more functional type of measurements in the brain, particularly, until let's say the beginning of the 20th century, when we started developing EEG, the only alternative we had to do measurements on the function of the brain was to perform invasive measurements, such as this one that I'm bringing in this photo. What we have here in this photo is basically an open skull. What you see here is what we call ECOG, or electrocorticogram. And ECOG is a measurement in which you're detecting electrical activity, just as you do with EEG. But now your electrodes, which are these silver colored points here, each one of these are one electrode. So you basically have a grid of electrodes spread in a specific part of the brain. But naturally, to do that, you'd need to open the skull. So you'd have some kind of invasive measurements. fNIRS, fMRI, and all these other techniques, which we call functional neuroimaging, they are placed in a specific, they have a specific role, which is to non-invasively understand or investigate the human brain. And here are some of the techniques that you can do this. On the top left, we see fMRI. This is magnetoencephalography, which is an analog of EEG, but with magnets instead of detectors that can detect electric field. Magnetoencephalography, they detect the magnetic field. There are advantages of magnetoencephalography over EEG. The main one is the capability of localizing the sources of that activity, which we have seen today. The spatial resolution of EEG is quite low. So the idea of magnetoencephalography is to have the same type of temporal resolution as EEG, but with an increased spatial resolution. So you have a very good, from all the functional neuroimaging tools, magnetoencephalography is the one that has the best compromise between temporal and spatial resolution. It has an excellent temporal resolution, which is in the order of milliseconds, such as EEG, and it has a very good spatial resolution as well. What is the problem of MEG? The price. An MEG system today can range from $1 million to $5 million. The room that you need, which is a magnetic shielded room that you need to have your system placed in, there is one magnetic shielded room at MIT that costed $7 million, just the magnetic shielded room without the system that goes in it. This is the bad news, but there are good news, which is there are a group of physicists working in the US that have developed this very special type of magnetometer, which is an optical magnetometer, which is supposed, they're testing it already in cardio signals and in the brain signals as well. And the cost of these systems can come down to $200,000. So we have a chance in the next few years to have a very good magnetic detector, such as this one, costing $200,000 as compared to the $7 million that we have in the current systems. Here you have the EEG. This is transcranial magnetic stimulation. These are all the nuclear medicine tools, such as SPECT or PECT, single photon emission computed tomography and positron emission tomography. We have these two equipments here in Natal. The Liga Norte Rio Grandense do Câncer, for instance, they have a PET. And SPECT is a relatively cheap equipment that you find in most radiological clinics. And then here you have the fNIRS, as one was presenting, a very interesting system, particularly, in my opinion, to work with children. So this is the biggest advantage I see behind fNIRS as compared to other techniques, because as Juan mentioned, is not as affected by movement as it is fMRI, EEG, and every technique is very sensitive to movement. In the case of fNIRS, there's no sensitivity to movement, so that becomes then a very interesting technique to work with children. So my idea here is then to bring the underlying mechanism through which you're able to detect these signals, being with fNIRS or with fMRI. So what we're trying to do here is, naturally, if you're talking about understanding brain functions, what we're trying to do is to detect neural activity, because as we all know, brain functions is a result of this very complex connection of billions of neurons, each one connecting to 10,000 other neurons. So this complexity of connection makes a logical system, so to say, that is capable of doing all these amazing functions, all these amazing cognitive functions. So the idea here is to understand how is it that we can then detect neuronal activity without actually looking at electric fields. This is what fMRI and fNIRS does, which is so what we're doing to detect the signals that originate on fNIRS or fMRI is an epiphenomena of the electrical activity of neurons. So what I have here in this slide is a diagram in which what we're looking here in this line is time. This is potential of the neurons. So you see that as time passes, we do have a fluctuation of these neurons, of the potential of these neurons. And these fluctuations, they're called excitatory post-synaptic potential or inhibitory postsynaptic potentials, which is basically the connection. What we are doing here is measure the potential on a specific neuron that connects to many other neurons. So what we're seeing here is the fluctuation of these potentials. So this is what happens in every neuron. It's always receiving information from other neurons, and receiving information means that the potential, the electrical potential of these neurons fluctuate. So what we see here is that at a certain point, when the neuron reaches a certain level of membrane potential, something very weird happens, and this is what we call a action potential. So what I'm calling the weird thing is that when the neuron achieves a specific threshold, it propagates an electric signal to other neurons through this process that we call action potential. So what we see here is that, in fact, a lot of the energy that our neurons spend, they are spent in trying to restore what we call resting potential. So in other words, a neuron, it tends to stabilize its membrane potential in something around minus 70 millivolts. So what you see here is that about 47% of the energy that neurons spend, they do spend trying to reestablish the resting potential after the neuron has fired the resting potential after the neuron has fired in actual potential. So after something like this happens in a neuron, and this happens in the neuron all the time, so neurons are always firing, which means they're always communicating with other neurons. What we are seeing here is that about 50% of the energy that we spend, that our neurons spend, they spend trying to restore the resting potential when they fire an actual potential. Another 34% of the energy that's spent by neuron is spent by trying to restore these small fluctuations that we see here as inhibitory or excitatory potentials that the neurons receive. So see that around 80% of the energy that is spent by neurons is spent by trying to restore the equilibrium of these neurons. So we can understand these excitatory or inhibitory potentials and the action potential as phenomena that are always destabilizing the neurons. So the neurons are actively trying to restore the equilibrium that the neuron has before, which is the resting potential. And you see that a lot of the energy that we spend in our brain, almost 100% of the energy that neurons spend, they spend trying to bring the system to the equilibrium after it has all these fluctuations, IPSP, EPSP, or action potential. All right? So the question here is, then neurons, they or the brain, it's not a source of energy. Where do cells bring their energy from? So most of the energy that comes to cells, they come from a oxidative metabolism. So everyone remembers the Krebs circle, for instance, which is the circle through which we are capable of producing ATP from glucose, for instance. And that metabolism, we need oxygen. So basically, the fuel of neurons, they come from oxygen and glucose. But our brain does not produce neither one of them. Doesn't produce oxygen, doesn't produce glucose. So where is it that the brain, where does the brain get the energy from? From our vascular system. So the vascular system is the one that's responsible to bringing energy to the brain and then bringing energy to the neurons so that they can use that energy, most of it, to restore their actual potential, to restore their resting potential. So we do have a very complex and very interesting vascular system, which I'm depicting here pretty much. So the vascular system, as we all know, it's composed of two main systems, the arterial system and venous system. One of them is carrying, so the arterial system is the one that's carrying the blood with oxygen and with glucose. So what happens in this system is that once it enters the cortical surface, it enters as an artery, and then it diminishes the diameter of the vase that's conducting the blood. When it diminishes the diameter, it becomes what we call arteriole. Arteriole is an artery which with a much smaller diameter, and with a very interesting property, which is the presence of smooth fibers. And the presence of smooth fibers in the arterioles, they promote what we call vasoregulation. So vasoregulation is a dynamic process that happens in our vascular system, which is capable of basically increasing the diameter of the arterioles or decreasing the diameters of the arterioles. So we do have a flux control. As a matter of fact, a lot of patients, this is one thing that I've worked for a while, which is the following. When you have a patient with a carotid stenosis, which is an obstruction in one of the carotids, if it has that obstruction for a very long time, you decrease brain reactivity because the influx of blood is very low in the brain. If you have patients then with carotid stenosis, the treatment for these patients, you have two basic treatments. One is a stent. So you insert a stent and then you inflate the stent inside the carotid. Or you do what is called endarterectomy. And endarterectomy is basically a surgery in which you open the carotid, you remove the plates from the carotid, and then you close the carotid, it's free to go. What happens in a lot of these patients is that they follow with a stroke. So you remove the obstruction, and then the patient just evolves a stroke because they have an impaired vasoreactivity. So in other words, you increase the blood pressure in the specific part of the brain. The brain is so used to have a very low pressure in that part of the brain before you had the surgery, and then the patient has a stroke just because it doesn't have that ability to vasoregulate the pressure. What our vascular system tries to do all the time is to maintain pressure. So this is it is always vasoregulating in order to maintain a constant pressure. This is what our vascular system is always trying to do, and the way it does it is by regulating specifically on the arterioles. which is the only place in our vascular system that has smooth fibers, that has fibers capable of contracting and dilating. So all the other parts of the vascular system, they are passive. So it's just like a conduction tube. It doesn't really vasoregulate. So once you enter then the cortical surface, you have the arterioles, and then after the arterioles, what you have are the capillaries. The capillaries are very thin structures in the end of our arterial system, vascular system, and it is in the capillaries where we have the exchange of all the metabolites that the neurons need. So oxygen, glucose, they diffuse on the capillaries. So it means that then you have an oxygenated blood flow that comes into the brain in the arteries. Then the diameter diminishes. It increases the number of branches, so it becomes very diversified or ramified in the brain. And then you have the capillaries, which is where you have these very thin type hair conducting blood, oxygenated blood, and then doing all the exchange for the neurons. After that, then you have the venous system. Then you have the venules and the veins, which then go back to the lungs. So this is pretty much how we bring energy to the neurons whenever they need and wherever they need. So the first intuition we could have is that if we have that mechanism, then what the brain could do is simply watering a whole garden for a rose. This is the metaphor that a lot of people use. So in other words, we could think that our nervous system would be organized in such a way that whenever you have a specific brain area in need of energy, we could think that we would have a very general and broad mechanism in which you'd have an increased blood flow in the whole brain just because a specific part of the brain is in need for that for energy, for instance. What we are going to see is that the mechanisms underlying this type of phenomenon is a lot more complex and a lot more subtle in many aspects. And these aspects have been first investigated by Roy and Sherrington, and together with William James. William James, as you might know, is a psychologist, so he did not ever perform a experiment. So what he has here is more of an intuition, which is, as William James, he had a lot of very good intuitions on a lot of stuff. And here he had a very interesting intuition on these vasoregulatory processes. Roy and Sherrington, actually, they've measured it. And what they see is that in these phenomena is that if you have a specific part of the brain in need of energy, that specific part of the brain is going to vasodilate. So you don't have a general vasodilation of the whole brain if one part of the brain is in need. What you have is that a very specific part of the brain gets dilated if that part of the brain is in need for oxygen and glucose. This is one experiment showing this evidence that I'm commenting here, which is the following. What you see here is the diameter of the arteriole. What you see here in black is a specific stimuli that happened in a very specific part of the brain. So what you see here is a tip of one electrode detecting activity from this region of the brain. What you see then here are the arterioles. This is what you see is the arteriole that actually irrigates that part of the brain. So in this graph, what you see first is that once you have a stimuli, if that part of the brain responds to the stimuli, you have a very fast increase in arteriole diameter. that happens together with an increase in blood flow. So what I'm saying is that there is an increase in blood flow in specific parts of the brain when that part is in need for energy. That is a very low, this is crucial, because if it was not localized, we could not do fMRI or fNIRS. So it's crucial to know that these hemodynamic changes that happens in response to a specific event, neuronal event, it happens locally. You can't see these numbers, but I can translate these numbers to you. What these numbers are showing, I'm sorry, the quality of the slides, when we transformed it to PDF, it diminished a little bit. So, but what we're saying, what these numbers here are showing is that the greatest increase in diameter of the arterioles happened closer to where the stimuli was coming from. So in this part, for instance, you have an increase in about 15%, while here you have an increase of 0.7%. So you see, and the distance from here to here, from this place to the source, is just 3 millimeters. So from this you can contemplate the idea that the changes in hemodynamic response, they happen in a very specific place. Here I'm talking about 3 millimeters. So what I'm saying is that if this arteriole that's 3 millimeters away from this arteriole will dilate 0.7% with respect to what this arteriole has dilated. So which means that the increase in blood flow is local. This is what's the most important part for now. So what is the brain structure? That there are many different substances and processes that regulate our circulatory system, our vascular system. This is one of them. So a very one of the candidates to do this vasoreactivity is one type of glial cell, which are the astrocytes. Astrocytes here in this picture is what you see here in blue. So you see that an astrocyte connects neurons to the capillary bed. So it means that the information of which area is in need for energy comes from the neuron through the astrocyte, vasoregulating the capillary bed or the arteriole that's close to that neuron. See? Clear? All right. This is the first experiment that an Italian, Rosso, has proposed. So the idea here was to see, well, if we have an increase in blood flow in areas of the brain in which you have an increased brain neuronal activity, it means that if someone has a stimuli, for instance, a visual stimuli, that's going to increase the activity of this part of your brain, which is the occipital pole, the first area in which visual stimuli is going to be processed. So it means that if you have an increased activity on this part of the brain, from what we are seeing till now, there is also an increase in blood flow in that part of the brain. So what this guy thought is, well, if there is an increase in blood flow in that part of the brain, that means that the volume of that part of the brain is going to increase. That means that the weight of the brain is going to increase. So this is what the guy had, someone on a scale like this. And then you'd have a visual stimuli, and he measured, this paper was published, and this is the data of the published paper, showing that indeed, when you have a specific stimuli, you have an increase in blood flow, and that blood flow will increase the weight of your brain. So he measured it in a very non-specific way. So increased electrical activity of neurons, increase of blood flow in that region. This is what we've learned so far. All right. There is the second piece of evidence. This first piece of evidence is what we need to do fMRI and fNIRS. Now I'm going to bring you the second piece of evidence that allows us to do fMRI, not necessarily fNIRS, which was a discovery made by Linus Pauling. So Pauling, as any great scientist, contributed in chemistry, in peace, and in fMRI. What he discovered, what Linus Pauling discovered, was that depending on the state of oxygenation of hemoglobin, you have a different magnetic property of hemoglobin. This differences in magnetic, so if you have hemoglobin or deoxy hemoglobin, what you have is a difference in how an iron atom, that's the one that's right here in the center, if you have oxygen, that iron atom, it will be shielded from the environment. If you remove oxygen, that iron atom is not shielded from the environment. So as we all know, iron has a very profound impact on magnetic field in general. We know that iron glues to magnets, for instance. So it gets attracted. So there is a very important magnetic property behind iron. So what you see here is basically, depending on the state of oxygenation of hemoglobin, you have a different magnetic property of hemoglobin. Oxyhemoglobin or hemoglobin, it's diamagnetic, while deoxyhemoglobin is paramagnetic. Those names doesn't matter. The only thing that matters is to understand that the states, the magnetic states of the hemoglobin changes depending on whether or not it carries an oxygen. All right. So what we see from a vascular perspective is that if you have when is it that you have oxyhemoglobin? When you have an oxygenated blood, mostly. Then where is it that you have deoxyhemoglobin? Is when you have that oxygen from hemoglobin getting diffused by the vascular system, getting diffused by the capillaries, and then getting used by the neurons. So this is where we have a change. So what I'm trying to say is that together with the increased activity of neurons, that you have an increased blood flow, that blood flow that's entering that region is an oxygenated blood flow. Which means that when you have an increased activity of specific parts of the brain, what you end up having is an increased blood flow in that part of the brain, and what you end up having is an increase of oxyhemoglobin in that part of the brain. Which means that you're changing in a very subtle manner the magnetic properties of regions in which you have an increased activity of neurons. Is it clear? So this is why I'm calling fMRI and fNIRS an epiphenomena of the neuronal activity, because actually what you're looking at is not neuronal activity. What you're looking at is the epiphenomena behind neuronal activity, which is the increase in blood flow, the increase in oxyhemoglobin locally, and the change in the magnetic property of that part where you have an increased blood flow. Is it clear? Questions? No, it's a lot of information, I know. So this is what people then did with MRI, was to have oxygen. Oxygenated blood with different levels of oxygen in the oxygenated blood. So they basically had a number of samples with different amounts of oxygen in that blood. And what you see is that indeed, in the MRI, we can detect these changes in oxygenation. So it's, of course, it's not my objective to have you understand this, but what we have here in detail, so what we have in this axis here is the oxygenated blood, and what we have here is the MRI response. So the only message I have here is that the MRI response, which is in this axis, varies with the amount of oxygenated hemoglobin that you have in that blood. And this is what we used for fMRI. So this is the first experiment performed with fMRI in 1992. Actually, there were 3 experiments performed in the same year with the same idea. There's one piece of, because all those findings that I showed you, Roy and Sherrington, that's from late 19th century. Pauling, 1930. So why is it that we waited so long to do an fMRI based on these phenomena? Technological challenges. So, and the main technological challenge is the following. If I want to measure these changes in oxygen level, I have to be able to detect that same region of the brain sequentially. So I have to detect it now, I have to detect 1 second later, I have to detect 2 seconds later, so then I can map the changes that are happening through these processes that I've just described. This process is what we call blood oxygenation level dependent contrast. So in other words, so blood is a name that gives rise to what fMRI measures. So blood is a contrast. What is a contrast? It's something that stands out in terms of brightness, for instance. So in the MRI, what you're seeing is that the brightness of that region changes if it has more or less oxygenated hemoglobin in that part of the brain. What was it that people did in the first experiment? What was the technological challenge that we have with MRI? The possibility of acquiring a image of your entire brain in 1 second. This is not trivial. This is very difficult to do. It actually rendered a Nobel Prize. So it's so difficult to do that a person, two guys, Lauterberg and Peter Mansfield. Peter Mansfield died 2 years ago. Lauterberg is still alive. One is American, Lauterberg, and Mansfield is English. So basically what they have done was to create a specific type of sequencing in the MRI that allows you to detect or to acquire the image of your entire brain in 1 second. This is one sequence that's called EPI, echoplanar imaging. If you do echoplanar imaging, what you have is a very good temporal resolution, 1 second, but a very bad spatial resolution, 3 to 4 millimeters. But bad in terms of MRI. So it's like all the energy you're putting into having this image being acquired in a very fast way, paying the price of not having a good spatial resolution. All right? This is what we have now. So if we go in the MRI here in the hospital, we'll have a special sequence that allows us to do fMRI. This is one of the reasons why fMRI is so widespread in terms of a technique being used for cognitive neuroscience. It's because we can use any MRI system to do an fMRI acquisition. We don't need to buy a specific system, as we need to buy fNIRS, as we need to buy EEG. You don't need to buy it. In any system that we have available today in any hospital, I wouldn't say any, but most of the hospitals, you'd have the capability of doing fMRI. So that then allows us, which is exactly what we do, to use the MRI of the hospital. So the MRI costs around $2 million. Naturally, we wouldn't have $2 million here in Brazil to be invested in research in that sense. So, but that I can use. So I can use the system in the hospital to do my experiments. So this is to say that for fMRI, you can do in any system. And this is the main reason why fMRI has been so widely used, because it's easy access to a very expensive system. So what was it that they did? They basically used this sequence, which allows you to detect your brain every second. And they did a sequential acquisition of the brain for 5 minutes. So in 5 minutes, they acquired one image. In this case, it was a little bit longer, but let's say that in this 92, you were able to do it every second. So what they were doing is to acquire one image of the brain every second. And then for periods of time, say for 30 seconds, they would flash a flashlight in front of the individual. So the individual would see a flashlight. It would have 30 seconds without the flashlight, 30 seconds with the flashlight, 30 seconds without the flashlight, 30 seconds with the flashlight. This was the protocol. So from what we learned so far, what is going to happen in regions of the brain that responds to that stimuli? When you have a flashing light, what you have is neurons that process that flashing light, increase its activity. So say in this part of the brain that is processing the primary stimuli, the visual stimuli, is going to increase the activity of this part of the brain, increase the electrical activity. Increase the electrical activity, increase in blood flow. Increase in blood flow, increase in hemodynamic, in oxygenated hemoglobin. Increase in hemodynamic oxygenated hemoglobin, you have an increase in the signal, in the MRI signal, you change in the magnetic property of that region, change in the signal that I could observe with the MRI. All right? So think what happens sequentially. If I'm getting one image at 1 second, this is what you have in this graph here on the top. So this axis, you have time. These bars here, they represent the periods in which you had the flashlight off, the flashlight on, flashlight off, flashlight on. So what you see here is that when I turn on the flashlight, each point here depicts the brightness of a specific area that responded to stimuli. Okay, so say that I have one area here in the brain that responded to stimuli. What I'm mapping here is the brightness of that area. So every second. So look at what happened 30 seconds when I turn on the flashlight. What happens is that the brightness of that region that responded to stimuli is going to increase. Why is it going to increase? Because of the bold response. So this is the bold contrast, which is the increase in contrast in brightness in areas in which you had an increased neuronal activity. Clear? So look at what happened. When you turn on your flashlight, increase in blood flow, increase in activity, increase in brightness. Turn off the flashlight, decreases. Turn on the flashlight, increases. So this is what you do with fMRI. This is the other paper published in the same year, proposing exactly the same thing. So you see here the same type of results. You have the lights on, you have an increase in signal. You have your lights off, decrease in signal. Lights on, increase in signal, and so on. So what is in a few words, what is the bold contrast? You have, we know that the neuronal activity is related to blood flow. You have an increase in oxygenated hemoglobin, and on the other end, you have the changes in brightness that you follow in the MRI. So these two, this is a typical exam. So unfortunately, there was a video here that you could pretty much understand how we do it. So what is a typical fMRI protocol? Basically, what you do is to interleave periods of rest with period of the specific activity or the specific cognitive function that you're trying to map. In other words, the example I gave was with the visual system. I was stimulating the visual system. But I could stimulate any other system. I could stimulate the motor system. How? Well, I have 30 seconds with the individuals at rest, and then 30 seconds I'll ask the individual to move their hand for 30 seconds. When they're moving their hand for 30 seconds, what's going to happen? The motor cortex is going to be activated by this, increased blood flow, increased activity, increased blood flow, increased signal. When I stop movement, decrease blood flow, decrease signal, decrease movement. And then I can use it for any function. I can do language. How do I do language? I can do expressive language. So as a matter of fact, this week we did a patient for language. So what is it that we do? We ask the patient, for instance, when I say the letter A, you're gonna mentally produce the largest number of words that begins with the letter A. And then I'll ask the patient to keep producing words, so verbal fluency, for 30 seconds. And then I ask the patient to stop. Think of a whiteboard, for instance. Rest. 30 seconds of rest. And then letter F. Produce the greatest number of words with the letter F. We're going to produce. So what's happening there? I'm recruiting the language network. Emotion. I can ask the individual to look at a aversive picture against a neutral picture. Of course, when you see an aversive picture, for instance, a car accident. So you see a picture of a car accident, that regulates your emotion, of course. So what you have is then the stimuli is whatever you want: memory, attention, emotion, motor system, the sensory motor system, whatever. This time perception. I want to understand time perception. So all I need to do is to change what I'm calling task for the specific brain function that you're interested in mapping. This is what we do. So what type of application we have with fMRI? The main clinical application is this, which is to do what we call preoperative mapping. So preoperative mapping is to have individuals in an MRI scanner before they do a surgery. So the objective of these type of mapping is to give the neurosurgery the location of areas that the surgeon should avoid. Because, for instance, this is one case. So you have a patient here. This is the lesion. So you can see here the lesion, and there's this brightest spot here in the brain. This patient was going to undergo surgery, and this lesion is very close to the motor system. So what we do is, before surgery, we have the patient going in the MRI. We have the patient performing a motor task like opening, closing hands, or feet. Sometimes feet is more represented here. So if the lesion is close to feet, you have the patient moving its feet instead of having the patient moving the hand. You can do a homunculus if you want, doing different fingers, doing different parts of the body, whatever. So what we are seeing here with this colored image are the brain areas. So this is a typical fMRI result. What you see is the anatomy, and on top of the anatomy, you have these colored spots, these hot spots. These hot spots were the ones that responded to the task that you had the individual performing. So this is the functional region that the surgeon is trying to avoid. You can do that for clinical evolution, for instance. So you have someone that it's like this patient. She had radiotherapy treatment, and then she had to go for surgery. She had 3 surgeries. So we could do fMRI progressively, because as you can see here, the motor cortex of the patient has changed significantly after surgery. In epilepsy, in memory, so memory mapping for epilepsy, language mapping, such as this case. So this ends up to be a very important type of protocol. Specifically in this is one case that I always like to bring. This patient here that you see on top, so I'm about to finish, but this patient here that you see on the top was a patient who had a stroke when he was 3. And then he returned to the hospital. Back then he had 17 years old. In this image he was 17 years old. He started presenting seizures, epileptic seizures, and apparently the source of the seizures were these frontal area that still was not was still preserved. So the surgical procedure here was to do a spherectomy, which is to disconnect the two hemispheres. What is the issue? That this is the left hemisphere. Most of us has language processed in the left hemisphere, especially if you're right-handed. So there was a chance that the language of these patients had migrated to this region. And then you do, and the procedure, you free the patient from seizures, but then the patient is not going to be able to speak nor to move. So what you do in fMRI, then this is the fMRI of the patient. So what we're showing here is that for this patient, language is in the other hemisphere. So this for surgery is very important because now he knows that after surgery, the patient won't be aphasic. So then this opens every perspective that you can think of. This is very complementary in one sense to fNIRS because what you're seeing here is the same kind of epiphenomena that fNIRS see, but with all the positive and the negatives of these two techniques, at the end, they're very complementary. All right, so this is it. Thank you, and I'm open for questions.
We are open for questions. I would like to start with a question, Wama. And I would like to know how the cold would be affected by hunger.
By
Hunger.
hunger?
You will be hungry.
Okay.
And the time of the experiment, like it's going through the lunchtime and some kind
Yeah,
of
so as far as the bold response, that shouldn't be affected. So you don't have any influence on the mechanism. Of course, that on the psychological mechanism, then you have an effect. So you shouldn't have someone hungry in the MRI asking the person to perform a test. But in terms of the physiological mechanism, you won't have an effect. You have, though, an effect from caffeine, for instance. So caffeine is something that influences significantly the bold response, as many other substances. But caffeine is so widespreadly used that we usually ask the individuals not to have caffeine a few hours before the trial, just because we know that there is an influence of caffeine and other substances.
Another question. So, the fMRI isn't a good technique for measuring long-term procedures or not? Like
It is.
a long stimuli.
So I guess okay. So yes, it is. So the fMRI has the advantage over other techniques, which is the same thing with fNIRS, which is it's innocuous. So there's no harm that you're gonna have if someone stays in the MRI for 3-4 hours. Yeah. But on the other hand, it's not a comfortable environment. So typically, an fMRI experiment, we try to keep it as short as less than 1 hour, because after that, it's just you just feel too tired. Especially because in most of these experiments, you need the collaboration from your volunteer because you're asking the volunteer to do something to perform a task. So one thing that's fundamental in these techniques is to make sure that the task is being performed well. And this is why in a lot of these exams, we need a feedback from, not a feedback system, but a feedback, say you respond to something in the middle of the trial, just make sure that you're responding it correctly, that you are awake and things like this. But it's related to your question. One thing that's clear is that the fMRI is a very interesting technique if you want to evaluate in a longitudinal way. So if you want to say you have a procedure today, for instance, let me see if I have it here. This one. So this is a study that we did with physical therapy. So understanding the influence of different physical therapy strategies on patients with stroke. So in this case, we had patients that underwent a functional a series of exercises, and the other group did not. It went to a non-functional group of exercises. To do this, you need an interval of about 1 month between assessments. So for this, the fMRI is really good. It's very stable in a sense. So the EEG is not that stable. You have, for instance, the impedance of the EEG is something that's very difficult to regulate, and then have two acquisition that the signal to noise looks just like the same. For the fMRI, it's very in 1 month. That's okay. You won't have a lot of fluctuations in terms of calibration of the equipment and so on.
Any more questions?
Thank you for the talk. It was great. So as I said, everything we measure here and with fNIRS is a phenomenon. So it's a very indirect measure of neuronal activity. So my question is, when we're using, especially when we're using the classroom fNIRS with naturalistic ecological research, what caution do you think we have to exercise when we communicate these results? And because when we communicate to the educational environment and community, we see that the teachers, they are very friendly, they like this kind of results, but we also don't want to communicate the wrong idea that this is, right, panacea of explanation and solves everything. So, and do you think that combining behavioral research and behavioral data when communicating the results, do you think
that's fundamental? I think this is fundamental. So today, so fMRI, I think, is migrating from what we would call a modern phrenology, in the sense that we are localizing brain functions to specific parts of the brain. So at the beginning, we see very clearly that the papers on fMRI was basically a list, a table with 200 lines, showing each line one structure that was activated by a specific task, without really making sense of what's going on. So today it's very clear that if you want to do an fMRI experiment, it's fundamental that you have a behavioral task together with it to make sense of it. So one thing that you see often, or always in the fMRI papers that are published today, is you have the results like this one, and you have a correlation. So there's, I haven't. So it's always, you have this, and then you have the behavioral task, and then you correlate the activity of specific areas with your behavioral task. The best is naturally to have them being performed together. So in other words, to have the behavioral task being performed in the MRI. That's not always the case. So the other thing you can do also is to have traits as covariates in your fMRI. So personality traits, for instance, and then you have that as a covariant in your study. So yes, you touched a very important point, which is fMRI. So we as scientists, we are very cautious in how we report things. We never say this is like this. We say this is likely to be like this, right? But then you have the media, the press, that it's the one that distorts the information. And the fMRI has been a very interesting candidate for the press for distortions, because it puts together two things that, when walked together, very dangerous: beauty and ignorance. So the images are very beautiful, but no one really knows what's behind it. So it's very easy to sell it, and then to say, well, we found this, we found that part of the brain is doing this, and so on. And then we have a huge gap between the cognitive processes, because we're using this to try to understand the neural basis of the cognitive processes, right? But we don't really understand the cognitive processes. We really don't understand the cognitive basis. So then you draw conclusions that are very fancy, very interesting, but then you don't have a lot to tie that conclusion on. So I'd say that, behavioral task, being very cautious in being complementary also with different techniques, because Then if you have, in that sense, if you have an fMRI and an fNIRS, what you have the complementary results saying, well, I found this in the fNIRS, I found this in the fMRI, the basis of the signal are the same, so they are complementary in the sense that you're saying, I found the same thing, using two techniques, different moments with different protocols. So I'd say that this is one important part for sure. Yeah.
Thank you.
Thank you.
I would like to know about the events related protocol. For example, in EEG, we have very fast protocols. For example, with 6 stimuli per second, and with 5 phase stimuli in one protocol. It's possible to manage that
Really cool.
in MRI?
Yeah.
Or it's impossible?
I'd say that it is possible, but as you were saying, and as we were saying, what we are seeing here is the hemodynamic response. So that was what I was asking Juan, because in fact, what's limiting the temporal resolution is the physiology. So even though you have a very fast neural activity, you have to wait for the vascular system to respond. So, it's not a matter of the equipment. So it's not a matter of, so when we talk about the temporal resolution of the fNIRS, it's not a matter of sampling rates. Why? Because, I can, if I had an fNIRS system that I'm capable of detecting one image every 1 millisecond, what good is that? I have 1 millisecond, but the response, the underneath response, will take 2 seconds. So I'm like sampling every 1 millisecond. That doesn't matter. That's not where the temporal resolution comes from, because there is an underneath limitation, which is the physiology behind it, which is the same limitation we have on the fMRI. We are detecting hemodynamic response, and the hemodynamic response is slow. So you can detect, I could detect like 100 images per second. That doesn't matter. That doesn't do any good, because I'm going to be detecting 1,000 images per second, but those 1,000 images will be measuring the same thing because there's no response, right? So this is one point. There is a limitation, the temporal resolution of these two techniques, fNIRS and fMRI. They're bad because of the underlying phenomena that you're basing your measurement on. But then, can we try to improve the temporal resolution of fMRI? And then there are techniques that improve the temporal resolution, which is basically by doing a technique that's called jittering. So jittering is this. Usually, when we do an ERP, with a regular spaced stimuli. So every 1 second, every 200 milliseconds, whatever, you have a stimuli. What we do in the fMRI is that we do a jittering, in which you have one stimuli here, and instead of waiting for the 20 seconds until the hemodynamic response comes down, you have a second stimuli. You interleave some other stimuli here. So then you have a composition of this 2-3 stimuli, and that increases your temporal resolution. For fMRI, you have basically 2 types of protocol. One of them is what we call block paradigm, which is what you see in top. So the block paradigm, you have someone performing a task for a long time and then a rest for a very long time. For instance, 30 seconds, 30 seconds, 30 seconds, 30 seconds. And then you have these other protocols that are called event-related. So the idea on event-related is that you have a short task. So instead of 30 seconds moving my hand, 30 seconds without moving my hands, I move my hands for 3 seconds. I stop, and then I have a quiet period for 30 seconds. So shorter task periods, longer rest periods. Where do we use this? And you're a specialist on that. The amygdala, for instance. So we know that the amygdala is a structure that has a very high habituation. for stimuli in general. So if you have these protocols being run in the fMRI, block protocol, 30 seconds, that you're trying to see the amygdala, you won't see it because it's going to respond at the beginning of the task and then it's going to habituate. So then you need to move to this other type of protocol in which you have very short stimulation and then you remove the stimulation and have a longer period of rest. So there are possibilities, but definitely you won't get even close to what EEG or MEG can do in terms of evoked potentials. Yeah.
Thank you for the lecture. I'd like you to, I don't know actually how to formulate my question, but you commented about the stroke patients in which you assess the patients before and after physiotherapy sessions. And I'd like to know how different interventions may change the growth signal. For instance, in that case, probably the restoration of the areas affected by stroke would be an explanation. But in my case, I normally work with healthy individuals. So, for instance, with exercise, there's a lot of people showing some cognitive benefits from exercise, and also some changes in fMRI assessed responses. So how does, for instance, exercise or physiotherapy sessions may change the body signal? Is it the intensity?
It depends. So basically what you look for when you're analyzing this type of data are two variables. The first one is the localization of the body signal. So for stroke, for instance, what we see, let me see if I find that slide. Oh, yeah, okay. So for stroke, what you see, so this is before intervention, this is after intervention, same patient. So what you see is that before intervention, you have a number of regions being engaged by the task. After treatment, you see that the number of regions and the concentration of the both response in a specific area, like the motor area, is a lot more concentrated here than before. So what you can see is patterns of activation. So pattern of activity in different parts of the brain. This is consistent with the following idea. When you're learning, for instance, how to play the piano or whatever, and if I do an fMRI, while you're trying to play the piano, while you're trying to learn to play the piano, you see that it's a difficult task, right? You're going to be very concentrated, and that's not going to be easy, and so on. And that comes together with a very spread activation in different parts of your brain because you're using a lot of your cognitive effort to do the task. After you learn the piano, what happens is that the activity or the spread of activation, they reduce, and you see more concentrated in the specific areas. So this is pretty much what we see here in this example. When you have a stroke patient that's trying to relearn how to move, it's gonna have this cognitive overload just to move. And then it's getting better with time, with the physical therapy, it's gonna get better. So it means that after physical therapy, that's gonna decreased amount of effort that the individual is paying to perform the task comes together with this less spread pattern of activation. So one thing is this. The second thing you could do is temporal evolution. So how is it that temporally that area responded to your stimuli? This is another thing that you can get from your results. And the third thing is the engagement of specific areas. So you, for instance, like in that same sense that you have a less spread from a more spread to a less spread, you also can analyze your data and say, well, before the exercise, you had this, and this area participating in the task. After the exercise, you reduce this area is not participating, and then you make sense of it. But that comes together with what Janaina was asking, which is you can't really do an fMRI just with the fMRI. You have to have a question a priori, and then you have to have a very well-designed behavioral task to make sense of it. It doesn't really make sense anymore to have a list of areas that responded to a specific task, saying, well, this is the list of areas that responded, like this modern phrenology that we spoke about.
Thank you. If we have an fMRI, it's good because we have big picture, so we can see all possible areas engaged in the task. But if you try to translate it to the fNIRS, for instance,
Sure.
we have limited channels. So
Yes.
let's imagine if I am assessing a patient with stroke or some clinical conditions, or even a health patient, for the same task. And I do an intervention between assessments in which I expect, in which my question is if I can expect a different magnitude of changes in the both signal assessed with
Yes.
fNIRS.
Yeah, sure. Yeah. I understand your question. Yeah, but definitely. Yeah, you're right. So, yeah, I think that the main point there is that for fNIRS, since you have the coverage is worse in fMRI, in general, it's advisable that you have an a priori hypothesis. So you're not looking at the whole brain to say, well, this is not the point. It's like, I'm going to focus in areas of the brain that I know it should respond to that specific stimuli. And you raised one point which I forgot to mention, which is the advantage of doing fNIRS with exercise, because as it is not something that's disturbed by movement. People here, for instance, in the Departamento de Educação Física, they have been working with fNIRS with that perspective of having people exercising, and while you have people exercising, you have your acquisition in place, which you could never do with well, you could, but it's a lot more difficult.
So I have one question here. And can we combine the EEG and the fNIRS?
Yes, you can. It's a very big technological challenge. You need a specific equipment that we have here. So we do have an EEG system that's compatible with the MRI. So you have the first part, it's a technical part. The electrodes have to be different. You cannot have your amplifier right next to your EEG. You need to have your amplifier a little bit farther away. You need optical fiber. You can't have wires. So there are all these technological difficulties. But the other one is more on the processing side, because in one end you have EEG, which is a very sensitive equipment for fluctuations in the electric field. And then you have the MRI, which is a powerful electric field generator. So if you look at the EEG inside the MRI, well, I should have brought that, but if you look at the EEG inside of the MRI, it's just noise. But the MRI is so precise that noise, you can model the noise, and then you extract the noise by knowing exactly how the MRI works. So there is a workaround. It's not simple to do, but it's possible. And yeah.
Any more questions? Also, thank you, Dráulio.
Thank you.
Great. And we are a little bit late on the schedule, but that's okay. Point here is to enjoy, and it was really enjoyable. Thank you, Dráulio. And we have the coffee break, so we'll keep the transmission, but we'll just nothing happening while we are at the coffee break, and we will return in around 15 minutes. and do the hands-on session. Thank you. So we are returning, and now we will start our hands-on section. Okay, we will demonstrate the equipment and show how to perform an experiment. Okay. Juan is going to start the hands-on.
Yes. Okay. Well, okay. So now we are going to start by showing the equipment. It's not that much, so we're going to take that much time. This is the main part of the NIRS. This is the amplifier. The amplifier has a charging option, the button here. It has an entry for the detectors, the sources. It has an entry for the USB on out that you're going to connect, which is the one you're going to connect to the computer. And it has a trigger option that is going to be connected to the manual trigger that I showed before. It would be this one right here, as you can see there. We have the detectors and the sources. They look quite the same. So here you can see that they are marked as D. They will be the detectors, and they always don't have any data. That would be the sources. Also, if you didn't have or the report, you will also see you will also will know that they are the detectors because the tip of the detector is plain, while the tip of the sources are round. Yes. And they also have different entries. The entry for the detector is smaller than the entry for the source, as you can see here. That it's made so that you don't have any issue connecting the sources and the detectors to the amplifier.
Keep on.
Okay. That would be the optics. You also have the cuts. In this case, we have a cup of 258. They are marked with colors and numbers. Those numbers are arranged. Those numbers and colors are arranged based on the 1020 system. The green ones are for the first 1 to 32 channels. The red ones are for the 32 to 64 channels, and so on and so on. These ones are good for the NIRS because you can place optodes wherever you want. And if you are going to use if you are going to make an experiment with NIRS and EEG, you can change the placeholders and place the holders for the EEG. It is quite easy. You just have to remove, and if you will remove the other one, and it's okay. So the configuration for this is quite easy. One of the issues with this mechanism is that this plastic can be broken. So that's kind of an issue with this type of parts. You will also going to have other cups. These are plain black cups. These are placed on top of the cup that you are going to place the NIRS optodes, and these are made mainly for tests. Do you want to come back this one? Yes. I think I didn't talk much about this part, but the main source of noise for the NIRS is the light. So when you are doing an experiment, you are gonna want that the room where you're doing the experiment is as dark as possible. If it's not possible, then you place these ones. These are just made for the light, so that the light can go through. And they work actually pretty well. If they don't work, you still have noise. You can place these aluminum caps that usually used in the hairdresser. They usually can work for this type of purpose of eliminating the light noise. Those are the caps that we are going to use. We are also going to use this this tool. This tool is made mainly for removing the optodes from the cap. We are going to see that in a minute after we place the optodes in one of the in one volunteer. These are important because usually the optodes from the NIRS are going to be really well placed, and to remove it and to take them off with your fingers is quite hard. So this can help you to do the process faster. You will also have two other options. This is a trailer cable. It's a LPT cable, AC to LPT. These are made so you can send a trailer from the amplifier. From the amplifier to the computer via LPT. And you also have the manual trigger. This will be the one that you will use if you're using a more realistic experiment outside of the laboratory. They can place one button and they will send the trigger to the software, to the register software. Then you will also have this.
Ask a question anytime.
Yeah, you can ask. These are made basically to place in the cups. Sorry. This one. These are made so that you can place them in the position you are going to place your optodes for your experiment. These are good because they are going to give you the certainty that the distance between the optodes is 3 centimeters. And are also good because you have too many options, and when you're placing the optodes in an experiment, you want that the setup is it's the fastest as possible. So if you have this in your cups, that will make the process of placing the optodes faster because you already know where to put them. They usually have many because they can drop. These are cable holders. These are placed in the back of the head, so you can place the cables of the optodes and the sources, and they don't get a string. They don't get they don't move that easy. And you have a meter. This will it will be the meter that you are going to use. The way that you're going to place the cap is the same way you're going to place a cap for EEG. You have to measure from the middle of the eyebrows to ceiling, and from the ear to the ear, and place the CC in the middle of the measurement of the senior linear and the ears. Last but not least, you have the phantom. The phantom is the tool that you are going to use to check the communication between the outputs. You are going to place the sources and the detectors here. You are going to put it in a really dark place, and you are going to check the quality of the signal in the software. If it goes well and everything is great, then the outputs are okay. That will be used only when you have any doubt that the sources and that the outputs are not working well. You can do it anytime you want. Usually we could do it maybe once per month or once per 2 weeks. That depends on the laboratory. But this is done mainly for that. I think that is it. You have also the cable from that you are going to use to connect the NIRS to the computer. This is the entry that is usually used in the brain vision equipment. The EEG also has this entry. I think that's everything that you would need to see. So I think the first thing that we could do is to check the silent quality, just so that you can see how that it's working properly. And how you can do that. So we are going to connect first the NIRS, the amplifier, and we are going to connect the sources and the detectors. Thank you. Um, um, in the phantom, what you're going to do is place the detectors in one place and the sources in the other place. I usually do it in order from the first to the last one. It doesn't matter that much, but I like to do it in order. Um, so you are going to place all the detectors here. Okay. In this test, we also want that the cables are not twisted. From here, this one, two, 4.
I have a question.
Yes,
The order that you are plugging.
it doesn't matter. I just like to do it in order, but it really doesn't matter that much. Because what the test is going to do is check for the communication between the detectors and the outputs. Now, we are going to open, can you share the screen? So this is the software that we are going to use. Its name is NIRS. All the softwares can be downloaded from the main page of NIRS. In the NIRS you are going to see different options. The first thing you need to see is that the instrument in use is the same that you have connected. It is possible that the device, that the software detects another amplifier. So in that case, you just have to close it and open it again. Here we have it already detected. That's the reason why you have to connect first the amplifier and then open the software so that the software can recognize it. It will tell you what is the source type. It's using LEDs. And the amplitude modulation is going to be calculated automatically. Then you have system setup, data visualization, and block average. System setup is going to be based on the layout that you are going to use for your experiment. Here, when you go to configure hardware and predefine montages, you are going to see montage, and you are going to see different options. Headband, motor, headband 8 times 8, motor 16 by 16, motor 8 by 4. If you use, these are standardized layouts for the placement of the objects and the detectors. If you wanted to use another placement, you will have to create a new one. I think because of the time, we can't make that today. But these are some of the options. If you open, for example, another one that has 16 by 16, for example, motor 16 by 16 is not going to allow you to open it because of the system that you are going to use. It's only going to allow you to open the ones that are possible in your system. In this case, 8 by 8. So we are going to keep the motor 8 by 8. If you were having another NIRS recording, if you are going to use super scanning, then you will press that here, but in this case we are not. Channel setup, this is going to be the number of the sources and the detectors that you are going to use. If you wanted to change that, you can, and so the detectors or the sources that you are going to remove are going to be deleted from the recording. If that happens, as you can see here, the sampling rate is going to change because you have less sources that are going to be sending the light. In that case, the sampling rate can be higher for the other sources that you have that you are going to use. You have the topo layout. This is right now it doesn't want to load. Channel masking, I actually won't move anything of that. Topographic display, the parameters. These are based on the modified Beer-Lambert law. So the modified Beer-Lambert law is the equation that you are going to use to change from light intensity to hemoglobin oxy and deoxy concentration. So in this case, for example, dPF is the value that you are going to use for the differential pattern factor, that is a factor that is going to take into account the age and how the light is going to be spread in the head after it is sending. This is important if you are going to use, if you are going to have research with kids, because the dPF is going to be different based on the age of the person that you are going to record, as you can see here. In that, usually the dPF is different based on the wavelength. The wavelengths that the system uses to send the light, it's 760 and 850. And the dPF is going to be that one. If you don't have, if you're not working with kids, or you don't have any theoretical basis to change the DPF, I won't change it. All these last things that you are using, that you are seeing right now, are things that usually you won't want to change.
Wow.
Yes.
So this is calibration process, right?
This is what?
Everything here is to test the quality of the signal, right?
Yeah. Yeah.
I'm trying to understand how the subject age makes a difference when testing the quality.
Now, in this case, no, I'm just showing you like the option from the NIRS start.
So this is already an option to record with not the experiment, right?
Yeah, this will be changing the recording of the experiment if you have any theoretical basis to change these values.
So when I'm recording an experiment, I have to put the age of the participant before.
Yeah, you.
Prepare the equipment.
Yeah. Yeah, you can do that. In that case, in my case, I usually wouldn't change that. When you're doing the offline analysis of the signal, you can change the DPF there also. These DPFs that you're going to change in the software is going to change how you're watching the signal during the recording. For the offline processing, you can change that DPF value again. I will change that only if I am working with babies. If I'm working with babies, then in that case I will change the age of the subject. Data streaming is going to be the way that an option that you are going to use if you want to stream your signal to another software. This is usually, in general going to be used because as we are going to see, we are going to do a demo with the NIRS team, that is the software of NIRS. The NIRS team is a software that it's quite, it's not that flexible to do experiments. If you want to make an experiment, you would usually want to use another software that allows you to make to do more presentation of stimulus more flexible. If you want to show two images at the same time or change the time that the image is going to appear, all of that. You are going to use other softwares. In this case, I use PsychoPy. And to communicate the NIRS with other programs, you are going to have you are going to stream your data to those programs, and you're usually going to use it with LSL. LSL is Lab Streaming Layer. Lab Streaming Layer is a program that allows the communication between different devices of neuroimaging. For example, if you're using EEG, NIRS, and PsychoPy, Lab Streaming Layer is going to communicate all those devices. Okay, I think that's all. So in this case, we selected motor, the motor montage. Sim. Yes. Now we want to test the signal. I'm going to one. Yes. Just to make clear, this procedure that you check the integrity of source and detectors using the phantom does not need to be before every single session that you are going to have with your volunteer or patient, right? Yeah, no. It's not necessary. It's only when, for example, when you get your NIRS for the first time, you want to see that the outputs and the sources are working properly. If you have any doubt that the outputs are not working well, then you will do this test. I'm going to do it here. Just to close it. And then here There is one issue with the window here. No, I think we don't need that. And because it doesn't fit to the screen. But here you're going to see the main functions. What we want to do is calibrate the signer. Here what it is doing is checking the quality of the signer. So let's see.
Let me see if I'm right. It tries each pair. It gets one source and one detector. Sends the light and see how much got on the detector. Then another pair, and there's that on every pair. Is that okay?
Yes. In this case, it got one wrong. It can usually be because it is not well placed in the phantom. But as you can see, the other ones are green and yellow. You can try it again.
Close the
And yeah, it's not the best.
This
setup
should
to
be
check
checked on a completely dark
in a
room.
completely dark room. Yes.
Yes, you can off the light.
Yeah, it will have some yellow ones because of the way we are testing the quality here. But as you can see here, the one that was red, it's already green. So this test can be done in a more in a better place. But as you can see,
Control environment.
in a more controlled environment, yes.
But overall, everything is green or yellow.
Except for those two ones that you will.
We are good to go.
Yeah, you're good to go. Okay, you can do that, check that test for any montage, occipital, prefrontal, and it always has to give green. Now that we are good to go, we can place the sources and the text
We're doing it next year.
in a scale.
Okay.
And it is
We need to have to get a subject.
in that subject, yes.
Usually these sessions take at least two subjects. One that everything works fine, and one that there are some problems, right? The main problem here is hair, right?
The main problem here is
There's a lot of hair. The hair may block the light, right?
yes.
You can do the experiment, but you will have
Not.
much trouble to adjust the electrodes.
Yeah, I think we missed to try some things to move the hair around.
To get away the hair.
Yeah.
Okay.
We would usually need something to move it. We don't have anything here to move it, and I think for the time, it's not that.
Yeah, because we are short on time, we may want to get one subject that might everything works fine.
Yes.
Okay. So I'm looking for one subject.
Yeah, perfect. Okay, but first let's just place the.
Can do some attention.
Yeah, he's the one. Okay, as you can see there in the screen with the montage, you guys can see that well. That here you have the numbers that you are going to have in the cab. If the cab is not new, it is possible that some numbers are missing or something like that. It is better to check it. If the cabs are new, you won't have any problem. So here, this green one is D1. That will be detector 1. The green ones are the detectors, the red ones are the sources, and they are based on the number. That's why you need to take care of the labeling that the detectors and the sources have. So you will place the red one that is in FPC will be this one here. Yes, so that is the front of it. Then you will have reference that this one, and based on that you can start to place the sources and the detectors. So I don't know if anyone wants to do this here or not. So here we place first the detectors. So detector one should go. Let me see this. One goes here. Let me just move this around. Hi? Yes, of course. If you want to take that. Okay, so the sources are going to be separated by two main cables. The first one and the other first. Yes. So you are going to place the ones that are here in this part. Here. But if you want to leave that here, and you can place the detectors with me. So these are the 5 to 8 detectors. These are the 1 to 4. So you have to search this D5, D6, D7, D8. So it says here D5 days, then it will be this one, just that it's broken. Now, yes, it's this one. And this. Yeah. Let me just maybe this. And then
22.
the 8. Okay, you have to search maybe. What? That one is here. I because it. Yeah. You do have more experience in this case. So let's. I think they are not orange. 12, 14, 15, 16. This 6. This is.
So I have a number 10 here, detector 5 is number.
Will be this
Number-
one.
So detector 6 is in number, the other side, here. Yeah. Detector 6 is in number 15, right?
Looks
20.
like a
20.
looks like a 12.
Yeah, the problem here is that the
I think it's this one.
Because the front is here.
it's a lot easier if you have a head model.
Let me
Maybe
just
we should
let's just do it in.
You
In his
feel a
head.
little bit of pressure, but nothing that will hurt you, I promise.
Yes.
When you see blood moving, so we stop that. Yeah. Can you remove your glasses, please? And get a little. I think that it's better if he turns to the audience. What's your name, please? Gilva. Okay. So we can, in this case, this cap does not have the EEG positions labels, just the numbers, but normally the system comes with a cap label. So it's a lot easier to identify visually. So one strategy that we normally use to place the cap is to identify some positions that we normally uh, use as easier place, such as CZ or CZ. But the problem is that CZ is only one point, so the cap can be like placed like this or like that. So I normally use FP1, FP2, and CZ. So I know that the cap is correctly placed.
In this case, we didn't measure the head of him just because we are doing a demonstration. But what you do, one of the ways is measuring from the middle of the eyebrows, assuming that is the bone here. And that measure, you are going to take 10% of that measure, and that 10% of the measure is going to be the height that you are going to need from the middle of the eyebrow to the first location. In this case, this will be FTC. Yes, that is the way that you're going to place based on the 1020 system, that is the one that we are using in this case. So here, this one is here.
And this is an old version of the system that uses a little row of holders. So the new system is a lot easier to plug and unplug.
Okay. So in this case, what we can see here, as you can see in the image, for example, D5 goes to 10 green, D6 goes to 20 green, D7 goes to 15 green, and D8 goes to 11 green. Yes, that's the way that you are going to place it. I was having some issues because when you have it without the head, the numbers get together too much. Like this is easier, but as I told, this is going to need some strength to put it inside, so it can hurt the person. Now, as you can see here, the string is pulling down this one. Can you see that it's going this way? I'll show you. Just that you can see that that can happen, and to avoid that, you are going to use these ones. So you are going to place this one far away from the place that you are going to do the recording, and then place that there. In this case, this one is actually like that. So, like this, you are going to avoid that the cables put down the detectors. As you can see here, that one is already okay.
How many channels do you have in this device?
So we have 8 detectors and 8 sources.
I did that.
8
Eight,
detectors and
okay,
8
yeah.
sources. Yes. So the easiest way to place this is to start with detectors and their sources, or the sources and then detectors, just to avoid issues with the cables. So first detectors and then sources, or the other way around is the same. So I'm just going to.
And it's possible to use the EEG, the electrodes
Yeah.
of each
So in this
at the same
case,
time.
for example, there are some EEG electrodes that can, I think they can be placed here. I don't know for sure because I have never worked with those. The new ones that are the Easy Cup, they have a different holder. It's more plain, and you have just to slide it. So what you will do in that case is remove this placeholder and place the EEG holder.
Okay.
And then you can place all the EEGs. And that's the way you are going to do the recording for EEG and NIRS.
But if you do that, you decrease the number of channels.
No, in that's another discussion because usually in this case you are always going to have a detector or a source that is going to overlap with a position for the EEG. Here you have to make a decision. What is the, based on your question, what do you prefer, time resolution or local resolution? If you prefer local resolution, then I will prefer to place the NIRS to check for that area and then move the EEG location around. Or if you prefer you have any EEG hypothesis more based in EEG, and I will place the EEG in the venturi system. Yes.
This is a cap with all the EEG positions labeled, and this is the new system to use with the sources and detectors. So instead of connecting with a holder that holds it in place, you use it in this new type of holder that is kind of loose. So you use this piece here and
Okay.
do like this.
Okay.
So this is how it looked like. So the mola, which I have no idea right now. Is what keeps it in pressuring the scalp.
Okay.
So it's just like this. As posições dessa touca estão bem estranhas.
Ai?
As posições dessa touca estão bem estranhas.
Sim, tem que ver isso.
Você quer que eu prepare essa outra rapidinho enquanto você vai demonstrando aí?
Tá bom. Ah, ok. 5, 8, 6, 5. Yes. Okay, so as you can see the positions here, it has the detector in the middle and 4 sources, and the other detectors are here. So the channels that you are gonna have, for example, in this case is the 5 with 5 and with the source 5 and source 6, The 7 is going to have a channel with 5, 7, 8, and 6. The 8 is going to have a channel with 6 and 8. So those are the communications that you are going to have in this case. And sorry. This one is not. This is okay. So here, what we're going to see is how to remove it. As you can see, it has a little thinner tip. So what you are going to do to remove it is place it down of the detector and pull it like this. It's that easy. So we are gonna have detector here. Source here. Detector here. Yeah, this is not properly placed. Then we are going to place detector. It's going to be here. One is going to be here. Let me see something. Okay, I think we are good to go. But let me just what? No fault.
How many channels missing?
Não, não, o problema é que o número não
I have a question.
igual que
I don't know if you already talked about it, but I saw in the software that there are reds and greens.
yes, source detector.
Yes, but here in the in the cap,
Yeah,
we have the yellows, greens, and
okay. No.
just
The color of the number is the color of the numbers that you are going to see here. Yes? The color of the field places, greens and red ones, are the colors that are going to tell you if it's a detector or a source. In the objects, you are going to see that it's a detector because it has a D, and it's a source because it doesn't have any letter. Yes?
okay. Thank you.
So all the channels are.
Now, all the channels are okay. The issue is that the number is not the same as here.
Okay, so
That's just one thing that we have to check later on. But right now, it should work.
But on the calculation, and on the output,
On the cap.
the cap.
Excuse me. Okay, so here we are going to place the black one. This is to avoid the light. And let's check if it works or not. So it's always going to be updating. Yeah, I do. Check it. Let's see. Okay, so yeah, I'm having some issues. I think it's because when you have issues, what you do is go to details and you are going to see what is the problem. So you have signal quality, gain, level, noise, and dark noise. So dark noise is the one that is going to tell you about the light. In this case, we have issues with the light. The noise can be,
The room should be completely dark, right?
yeah, and the level. So the issues are here, but I think it's because of the placement. Okay. When you have the signal, as you can see here, excellent, acceptable, critical, and lost. Excellent is that the signal is okay, it's communicating properly. Acceptable is that it has some noise, and it's analyzable when you're seeing the signal in the software. You are not going to see that well, but it is possible to record like that. Red one, it's now a problem in that case. Red one is it's a problem, and white is that it doesn't have any kind of communication between the um, the detectors and the sources. So here, what we are going to check first, it just it gives me some issue because you have to place it well, just that as because he doesn't have any hair, so he's going to feel the tip of the detector and the source there in the head, and that can be another
So that the those may not be
placed properly. Yes,
inserted, right?
yes. So let's check that out. And let me check and then there. T1, T4, S2, S1, S3, S4, S3. And this one is P7, P6. Let's try it again. Just to see if it works.
Principal aí, vamos fazer o fato, vamos. Que não é o dia que dá para funcionamento é inteirar a baixa.
We are going to place this. This XOR. This will help us with the light. And let's check how it works.
Melhor.
Yeah. So the signal improved. We still have some issues with that noise. That log of that there is that there is too much light around it. And that is a problem. But I don't think we have something around that. Which is we have that noise.
Oh, the monitor, right?
Right?
The monitor, right?
Yes. But sorry. Let's we have too much around. We have too much light around that's one of the light.
This was light.
You can see.
It sounds like one sign is better than the other, right?
Now, here when I'm going to press OK, it's going to go what? Um, it can also be that we are using the
Here is the nose, right? Here is the nose.
certain
So it's this light from here, right? Maybe. Maybe they're not. Then we can turn off the monitor, project the screen for a while.
Let's check.
Aceitabilidad. Aceitabilidad. Aceitabilidad.
It improved something. Okay, so let me show you this. Check this.
Maybe now it will. Yeah.
Right?
It's for the demonstration, right?
Yeah, but it's like it's fine. Yeah, let me, no, it's okay, just let me show, it's a demonstration. When you're going to do the experiment, then then in that moment, it's better to do those kind of things. But I want to show you something here before starting the silent. This, it can be the light, usually. But as you are going to see here, it's going to be green and then it's going to go yellow and white. The signal. Let me show you. As you can see there, it goes green and yellow, most of them. But then it says it's that noise. That noise is the one that is causing the signal to be bad. So that's the thing that, or when you're setting up the place to do the experiment and all that.
This is what you need.
Yeah, if you want, can you move?
Let's just rise.
So this problem is a common thing or
It's possible. You also have to think that the NIRS is not new. It's a news that it's usually used for trainings, and it has been moved around a lot. In a new NIRS, we usually have much better signal. You just place it and the whole thing goes green.
the thing that I was worried about the
Yeah.
place of the experiment?
But the place it's really important. Why? Because of this. If you are in a room without any kind of light, you're not gonna have this issue here.
Let's try it.
Let's start. Let's check the. Yeah, as you can see here, that the noise test is still, it's not that much light, but it's not green. So when it can be yellow in general is going to be red because of this. Yes?
Okay, question. But in an experiment, we always the light of the monitor. We always light
Yeah, that
in the room.
there, yeah, you will have this, but I think in this case we are having some issues because of environmental light. But those are things that you can work in while setting up the place to make the experiment. What I would do in this case, if I was in the experiment, is move to a really dark place and check for the light.
Check.
Just like this, just like the way we did with the phantom. The phantom was in a really dark place, and there you're gonna have this time. After that, you can,
One,
yes.
so maybe it is a good thing to, if you want to just send an instruction to the subject, say now do it, now don't do it, something like that. It be auditory instruction,
No.
not a visual instruction.
Visual can be also, but it's just this kind of light is not usually going to be an issue. It's going to be the environmental light.
Yeah.
When
We have a lot of light here.
yeah, because we can see each other. Just for that, you have light here. And then in other tests, usually the NIRS works with the side of eye and all that. You can have the person watching the stimuli in the screen. It's not a problem in that case. What I would recommend is a place without any environmental light. Just without the windows and all that. That's what I would recommend. Sí, sí, ya que hizo, ¿no? You can see the sign on here.
Maybe you see there's some green.
Yeah, there are some green signs. It's just not that bad. Just that the high noise level is too much. Because you would have this if you didn't have that. If you didn't have those big windows there, you would have a signal like that, or even better. But no, we don't have that right now. So, you want to see your signal? It's here. Yeah. We're just going to check the signal how it looks. After doing this, reading the signal, checking, let's imagine that it's everything green. You can check a preview. First, you will place the name, the age when you want to do that. And here you are going to see the signal. So here you can zoom in for the channels. You can change the scale factor. It is too big. You can change it to more small. Let's say here, or let's just go to this example. And then you are going to see the sign. You can change the factor as you want. The red one, I believe, is the oxy, and the blue one is the deoxy. As you can see, usually they are going to be populated. But here you will have like this movement of white, but it's going to be different from the blue one and the red one. Yes? That one channel is weird. That, for example, is a channel with noise.
The 2 and 3 are good, right?
Yeah, it is good. The 4, for example,
this up and down is related to
Yeah.
the heartbeat?
It can be related to that, but it's usually the oxy concentration in that area. You can move around. As you can see here, there is no change of anything. So that those are noisy channels. This is an example.
Show a very good change.
A very good channel would be the first one. It would be something like this. Because when you have stimulation, then what you're going to see, it's an uprise of the red wave and a downloads of the blue one. Just like the graph that we saw. Of course, it's not going to be the same, but that's the behavior that you're going to see after showing some stimulation. That one is just too noisy. It will get something like this.
It's a motor content, right?
Yeah.
So we do some motor testing now, right?
Yes. Okay, so let's come here. Okay, I can. So that's the preview. We are not recording anything. It says here that we are not recording. This is usually checking the signal. So you can stop this. And then you can see the signal. In general. If you want to, that one, for example, it's a little noisy, but it looks like a good behavior. And I forgot to show this. This one, it's something that I can load it for you. This is going to show you the activity in real time. So that's like the activity you can see here in the X. Concentration change. It's usually the same. It's pretty. Now if you want, let's try to finger tap the right hand. Just to see if we have any change. Look. Can you see? There is this change.
This hemisphere, the left hemisphere.
That's this is the left, this is the right.
Yes.
Is just finger tap the right one first, please.
The right.
The other one just leave it.
Yes. The left hemisphere controls the right hand, right?
Right. And as you can see some of the activity, even with all the noise. You can see that it can be seen the activity. Now please press the left one. The left one, I think it was with it had the highest noise. Can you press it more? Like this one going to go. Okay, so then you have the mark.
Okay, now again, right?
Yeah, it will be this one.
It's difficult to see in real time, right?
No.
To better see, you have to do the average.
Now the issue, yeah, that's true. But the issue is also that we have too much noise right now.
Yeah.
If you didn't have that much noise, we would be able to see it better. And yes, the average is also important. So what would you do? What you would do is to have, as you saw in the presentations before, the in between subjects of the or the trials are not usually that correlated. Can be different. When you average them and then you average the subject group, you're going to have the activity in general. Why? Because the noise is random in general. So you can have, because of the noise, more activity, less activity, but with the average of all the trials, you are gonna see the activity in general that you want to see. In this case, we could see that. Now, if you wanted to do the, I don't know if we have time to run the experiment. Do we have, how long do we have? Until what time?
Actually,
Okay.
but if was something short,
Yeah, usually, okay, so we can stop this.
yeah, okay, we can record something. Ah, okay, see, to record, right?
We can record something, just to show you. If you are not in a hurry, we can stay fine. But let's just, I just want to show you something.
Just do finger tap.
Yeah, just the finger tap. We can do 2 or 3 tries. You are gonna see there to press the left and then the right, just like that. When it says left, you are gonna finger tap your left and leave relax your right arm. And when it says right, you are gonna finger tap your right one. Yes. This is the software that I told you about, NIRS team. This is the software of stimulus presentation from NIRS. If you have a really basic experiment, you can use it. If not, then I would recommend you to go to other programs.
I have a question. Dráulio going to show us the fMRI and the paradigm of an fMRI, like test and
It's the same.
That is the same thing. Okay.
Yeah, it's the same thing because of that paradigm is based on the temporal response of the hemodynamic, the neurovascular process. Yes. So in NIRS, you're gonna have the same design. It's best. Now if you're gonna have EEG and NIRS, then you are gonna have to mix them in a certain way. This is the NIRS thing. It's the one that you're going to use to present the stimuli. You have introduction, then present left, press right. If you have the markers, marker 1, marker 15, those are the ones that are going to be sent to the software. And you can place them in here. So it says here that you're going to have introduction, then rest, then left, rest, right, left, rest, and so on. So this software communicates with the NIRS start. Okay, so I need something more. Sorry. You first want to start the recording. So now if you want to record, when you go to record, mark that X as X. Start recording. Okay, slower. And then we are opening the experiment. And I want you to see something. Okay. So here is the resting period. This is the resting period, the baseline period that you're going to have in the experiment. And then it's going to say that there you are going to finger tap your left arm. Can I start? There. Now here, as you can see, there is a dotted line, vertical. That's the trigger. Here. Yes. That's the mark to the recorder, to the software. As you can see, it will show the other trigger on the right. You are not going to be able to discriminate between triggers in the software, in the NIRS start. You're going to be able to do that in the NIRS lab. That is the one that you're going to use to analyze the data. We can do 2 more and then stop. So we have at least 2 minutes. So in this case, what you want is that the baseline period it's at least the same time as the stimuli period that you're going to present. So usually the stimuli period in here it's like 10 seconds, more or less. Let's see. Yeah, it goes from 120 to 100, right? Yeah, like 20 seconds, 15 seconds, I think. And then the resting period is going to be like 30 seconds, something like that. Because what you want is that the hemodynamic response goes back to the baseline period and then goes again to the stimulation period. And that's it. But as the hemodynamic response is that slow, then the coming back to the baseline period is going to be also slow. So if you are going to design and experiment with this, it's better to have that kind of designs. I think we can do this one and then close it just to show you fast how does the NIRS lab work. Okay. So let's just close it here. So we close it and then stop. And then we have this we can remove the yeah. If you want them
Really fast.
yeah, can you help me removing the card from here?
Okay.
Okay, so that will be the experiment. Now we will have to remove the card from here. You can just turn on the lights. While Daniel is going to remove the card from here, I'm going to show you fast the analysis. So to analyze the data, you're going to go to NIRS lab. NIRS lab is one of the options. You have many options. You can analyze the data in MATLAB, you can analyze the data in Python or in NIRS lab. NIRS lab is like the easiest way to do it, but it does have some limitations.
Is that fine? Yes.
Right? No, you can't remove it now. You can close this.
Right.
Find out that more and more coordination.
Yes.
Just different look.
Yeah.
It's already off, right?
Yeah.
Ja. Sicher.
Okay, so this is the NIRS lab. Right now it's in the 2019 production. So the first thing that you're going to do is load the data. There you're going to go to the folder from NIRS, go to data, and he is going to save the data based on the date. So here he saved the data from today. And we are going to check for the second one, that is the last one that we recorded. And we are going to load the configuration data.
The NIRS lab is a toolbox or not?
No, it's a standalone application. You can download it and install it, and it's going to work like this.
Okay.
It's built in MATLAB, but it can work without MATLAB.
Okay.
The probes are going to be loaded by themselves. You have to see that the imager system is the one that you use. In this case, use Parte. Then the markers. You want to go to the markers and set the duration. Let me check again. What about the duration from the markers here? So here, okay, so the trials have a duration of 10 seconds, as you can see here. So what we're going to do is go to NIRS lab, say that the trials have 10 seconds duration. Then you have to place here a duration, and then you can go to the event file and edit it. So here is going to tell you what is the order and the duration. In this case, the one I think it's left, yes. So you are going to say, if you have any question on which one is left or right, usually you can go to your presentation or to your software or your experiment and see which one was the first one. But in this case, it was the left, and then it went, no. Yeah, it was the left and then the right. So the condition 1 is going to be the left. So it's going to be left. The condition 15, that is the number of the marker that we have here. Then that marker is going to be the number of the condition. Marker 1 left, marker 15 right. Okay. So you can edit that. Left, condition 15 is going to be right. 10 seconds, 10 seconds, 10 seconds, it was free for everyone. And save and close. Okay, we can check again just to see it work. Left, right. You can see all the channels here. We have that. And that's now data preprocessing. The preprocessing is usually what you're going to do to clean your data. Then the processing is going to be the analysis of the clean data. So the pre-processing part is usually going to be truncated silence. Maybe you want to eliminate some part of the silence. For example, you were recording the data before the presentation of the experiment, and you recorded for, I don't know, 10 minutes before the experiment started. Here you want to remove that. In this case, we don't want to do that, so we can leave it like that. Interpolate. The interpolation of the data, I don't know how much you guys know about data processing, but interpolation of the data is basically average the channels around to have the channel that you had from the average of all the ones that were okay. But we don't want to do that here. We can check the data quality. As you can see, everything was okay. That's not true. You have to mark what are the criterions for naming as a good or bad channel. I don't know why it doesn't want to. It says it has one bad channel. I don't know why. That's weird. Here, this is the gain settings. This is like if this is in red, it's a problem because it means that the signal wasn't being sent properly. So we can leave it like that. The ones that are listed as bad are usually going to be marked in the software and then removed from the analysis. You can filter the data. Usually you will leave it as the default results. If you have any theoretical assumption to change it, you can change it. But here we can leave it as the default values. You can see how did it change the data. Yes. Now that we filter, we can save and close. You can remove this because we need these spikes, but that's like we don't have time for that right now. After that, you want to get the hemodynamic state. So now you have light. Now we want concentration, oxygen dioxide. To do that, the program is going to use the Beer-Lambert law. The Beer-Lambert law is the equation that is going to change from light intensity to oxygen dioxide concentration. Here you can change if you have another distance, the distance between channels, the DPF as I showed you before. You can change it here in the analysis. For example, if you were using the babies, you change the DPF for weight left 1 and weight left 2. Here we are going to leave it the same. This is the coefficient factor, molar extinction coefficient. That is the one that we already saw. That is the moments where the extinction coefficient is going to be highest. If you wanted another one, you can change it, but it's usually quite the same. So we can leave it as this one. Safe and close. We already said it, so we can compute the hemodynamic response. It has been, I think it already did it, so we can plot the data. Yes. We have the data here. It's like in general all the data around the experiment. It doesn't have the triggers. If you wanted to exclude that channel that you already know that it's wrong, because for example, when you were checking the data if it's bad, you could exclude the channels here. Now we want to block average the data. And this will be the block average. In this case, for example, we have oxy for condition left. Amplitude is set as the same. And this will be the response for condition left. Let me show you the two conditions. We want to replace the data. And there you will see a difference. Even if we had noise. This is a channel from the left. This is placed right, left. So in this case, for example, we had a good one because this is the presentation of the stimuli. This is the response for the right finger tapping and the left finger tapping. As we're seeing a left hemisphere channel, we can see that the right condition is higher than the left one.
This is the what?
This is
hemisphere. What is it?
the left one. Is this one?
Okay, then on the left, the right one is bigger, so it makes sense.
It makes sense. Yes?
Yes.
No? Yes. Okay. Good. Let's see if we can.
Because the left hemisphere controls the right hand.
Exactly. Here we got it. The other one here. But well, that's like here we have it again. I will check only the front ones, the frontal, well, the more frontal objects here, because we're the ones that were green check the quality of the signal. And in this one, it also contains the response. So it's working well. The response is the one that we were expecting.
Is it normal for it to be like
Yeah.
this shape?
Yes, it is now because you have a theoretical response, but the theoretical response is not always going to be the same as the preliminary response that you can see. This is a topomapping of the activity. You can see how it changes frame by frame, or you can animate the activity. You want to show them. Okay. Here we have activity there. The changing based on the.
This is the average, right?
This is the average, yes. Well, you can see that there. Here we don't have any groups, so we will leave it like that. We are not going to do that because we don't have groups. You can do the SPM, level 1, but this is the statistical parametric mapping for within subject analysis. You can load the NIRS info, save the place where you are going to save the OXI and the POXI data. You want to set the parameters for the ELM. This is like the statistical analysis that you are going to use. And here, well, you will have to check the way that Elidar says that usually you analyze the data. We are not going to do much things. We don't want to pre-whitening. To do pre-whitening, we will want to use this basis function. So just this part is quite hard and it's a really statistical part. The GLM, what the GLM does is basically take a beta. A beta, it's like I have a response, that would be the HRF, which is this one. And what I want is that what they do with the GLM is take your signal and compare it with this signal. With
The expected response, right?
Yeah, compare it with the expected response. As the signal is more alike to the expected response, it will have a higher beta. So for example, this is the expected signal and my signal went like this. That will mean a higher beta. But if it went lower, that will mean a lower beta. The beta values are the ones that you are going to do to make the statistical analysis. Yeah, no, but this is the statistical analysis that you need. So the statistical analysis for the NIRS data is basically applying a GLM. Yeah, the GLM is going to compare your signal with this and it's going to give you a beta value. You can do the whitening. Those are more advanced subjects that we can cover right now. That will take an entire training section just on analysis. Multiple conditions, you want the conditions from the list that five.
I'm going to finish.
Yeah. We're just gonna finish. So this is the design matrix. So it's going to tell you this is the time where we had the response. This is the activity that we want to see, and this is our signal. So what it's going to do is compare all of this and give you a beta value. This is the design matrix. So you have left, right, left, right. In different moments. Let's confirm that. This will be the matrix that is going to be used for the GLM, and we can estimate the GLM for features. And we can see the GLMs. We want to see the beta images. And here we have like the differences. So it says basically that they were different for the model one. I think this is the left one. So you will have higher activity in the right division for the left one. If you wanted to see the other model, you can check it, but I'm not sure how to do that. This is the beta one. You can go to the beta 2 and check it. If you wanted to see other things like SPM image, and you can specify the p-value and what. Here you already did all the statistical analysis. You have your data values. Then you can make the statistical analysis. You're saving the data, and then there was an issue.
Okay, but what is the plot of the beta?
What?
What the plot of the beta was telling?
What the beta is telling is, okay, I have
No, this plot.
yeah. In this plot, what it's telling you is the values of the beta for every one of the.
No, how do you interpret this?
You can't interpret the data values, but you want to interpret is the SPM image.
Okay.
That is the. I don't remember. We will do a T contrast. Let me just. The state gets trickier. Because here what I'm telling the software, and I don't know how to use exactly this thing, is give me the data values. Okay, let me just go here. The data values are the ones that are going to tell you. Let me see.
Let's convert the beta into a p-value.
Yes, you have the betas. So these are the betas for the left and the right for the left condition. Yes?
I see. And what is the range of the beta?
I don't know the range. That one I don't know. Yes, I don't remember what is the range of the values for the beta. I don't remember that. But it's going to give you a beta value for condition left. It's going to give you a beta value for condition right. And then you can use those beta values to do the statistical analysis.
Okay.
Yes?
Yes.
Only. Yes.
Yes.
Okay. Here I don't
You
remember.
need many subjects, right?
Yes. And you can do it in winning subjects. These are you have two conditions. One subject you can do and winning subject test analysis.
Yes. Okay.
But
When we show the plot, showing the average,
yeah.
that's bigger.
That's like the visual part. Yes. If you just go into the statistical part of taking the data, doing the
Yeah, it's like
yes.
Yes.
you don't use a normal, just use the
Yeah.
And that's how you will analysis. How you will do the analysis.
But one thing I learned in the training is that this statistics is a little different than what we are using with noise.
Yeah.
Because we compare the response with expected response. Right?
Which will be
And the data tells how close the response is to what the model expected, right? Yes, you're expected to rise like this. Right?
this statistical analysis is more alike to the fMRI. They do this with their response.
which is
with
the
their-
main division of the disease model.
What is what?
Yeah.
MATLAB.
What?
What is this program? MATLAB. Yes. I think that's it. That would be like
And we can enhance the function.
what?
I can use the function without interface.
Okay. Now, there I don't know. You can do. This is just one of the programs. You can go to, if you're using, for example, MATLAB, there are other programs like Homer or these tools.
And then you work inside MATLAB. This one doesn't work. This one was built in MATLAB, and then it was made as a software, so you don't actually need MATLAB to run it. If you wanted to have the function to analyze it, then I would recommend you to go to NIRS tool or somewhere that then you have the functions in the. It's easier to access the functions than the have. I think that is it.
So we have a short question from the chat. If you can do fNIRS to study epileptics.
I don't know.
Of course.
Let's start.
Yeah, I don't know. Sorry. I haven't actually read anything about this and epileptics. But I am not familiarized on what do you usually see in an epileptic study. Yeah. So I don't know that.
Okay.
Is it free software?
Yeah.
Yeah, it runs in MATLAB.
You don't, yeah, you don't need MATLAB to run it.
You don't need.
Yeah, you don't need that. This computer doesn't have MATLAB.
Is it free?
It is free. Yeah. There is another software from Python. Python has fMRI, and that it's a general program to analyze the imaging data. You can analyze the designers, image designers, and they did a library for analyzing this data also. If you don't have access to MATLAB, those are some of the.
And that other software to present this thinking, this software
It will
comes
be.
with the device.
You can sync this with Psychopy, for example. Psychopy is a free software that I think you can even use. It's easy to use. You can use any similar software that has integrated an upstream layer.
With triggers.
For the triggers, exactly. For the triggers and for communicating, for example, with EEG, they are really good because EEG and NIRS data have different sampling rates. As they have different sampling rates, the triggers get triggered.
Yeah.
Yeah. So what the upstream layer does is that it synchronizes those triggers based on the sampling rate of every device in the company. Yeah.
Great.
Yeah. Okay.
Any more questions?
Thank you.
So thank you.
Thank you, great support. Thank you.
Ela merece duas lancheirinhas. Ainda bem que ela superou essa câmera, né? Gente, more productivity.
Thank you. Great. So, thank you to all to everybody watching. And that's it for today. Let's see. Okay. So, finish the transmission. Bye bye.